Good, thanks. All right, I think this recording. Okay, so thanks everyone for hopping on. Today we got Cassian Yee.
Oh my God.
I
Where
found
did
your
you get
picture
that picture?
on
This
MD
is,
Anderson.
I can't get that picture off the website. Off the MD Anderson
You got to
website.
update it. Hey, this is what it is.
All right.
But I'll get an updated picture of you
No, it's
next
fine.
time.
It's fine.
But yeah, Cassian's going to talk about some really interesting data about SARS-CoV-2 epitopes. I had met him about a month ago or so at actually a NASA conference, and we started talking about some COVID and long COVID data, and then he said, "You didn't think this data you had was real," but then I was like, "Oh, this is very fascinating data here." So I'm not going to spoil anything else for anyone, but I think it'll be interesting. Other people will be interested to hear what he has to say, and probably this data cross pollination between our data and yours that we could join forces. Okay, so just a regular, no new papers yet, but there will be some probably. So for the variant distribution is now basically a JN.1. It's not just the US, which is the US centric, it's everywhere in the world. There is a couple of new variations of JN that I'll talk about. The wastewater in the US and the world is almost the same. There are, it's basically going down and plateauing a little bit here. You can see the regions. Biobot has some biases. Where I live has this spike here, but I'll go over that in a second. Sometimes these spikes are anomalies because certain regions outweigh other or compete. Like there's one region that might cause this anomaly, but there's other reasons this might happen too. I'll go into in a second. The CDC in the US is showing a low content, and overall it's low. So the CDC and Biobot have some discrepancies, but the overall average is about the same. Again, JN.1. But the trend is basically everything's going down, which is good to see, and that's worldwide. I've been talking about this guy, Wheelan, who has a really nice model to translate the wastewater into actual numbers, which is what we need now. And he's showing the same thing. One thing he talked about here, again, seeing a spike in certain regions, he talked about this is where I talk about it. He talks about to be cautious. There's another wastewater scan site we can go to where it says sometimes there's erratic data points that would cause these things. Depending on the location they do the wastewater, that would be causing the spike. But the overall trend you should look at in the state, and that's the key. Okay, so I'm going to go through it quickly, but JN.1, there is this new variant, JN.13, that's coming. It's a variant of JN.1, as you see here. And there's potential mutations here that might cause it to spread quicker, but no one knows if it's going to cause any other issues other than that. And boosters still protect against all the JNs pretty well, so that's the good news. And this is now just getting into the news. I was like Eric Koppel's blog here, but he's talking the 4-year perspective. I thought this was kind of interesting to look at, just to give you perspective. Was it a few days ago, or March 13th was the 4-year basically anniversary of the WHO declared, global pandemic, and basically the world shut down, if you remember. And 4 years seems longer. And that's how long almost we've been together. So that's kind of a 4-year anniversary of COVID is coming up. But yeah, you can see, as expected, the overall life expectancy since COVID has dropped, as you see it. But interestingly enough, the partisan line in the U.S., since elections are coming up, not to get too political, but you can see that people who were Trump supporters, and they've actually been killing themselves off for not following COVID guidelines. So maybe some hope with the election for depending on which way. Well, which way I was like. Okay, I'll stop there. But okay, so the evolution of viruses, as you can see, this is a nice thing. It's a nice little summary. He talks about JNs and JO and how the variations and mutations have accumulated, and briefly mentions JN.13, which is starting to pop up, has these extra mutations. But the wastewater, levels, as he says that. And one other thing that I agree with Eric, because I've mentioned this before, the CDC's policy towards isolation has basically they've given up now. Said that everyone's okay, you can go out and not isolate, but this is going to cause issues down the road, I think. So okay, let's go over some of the things that are papers and news. I'm going to go through it quickly just so it gives Cassian enough time to talk. So one thing is this paper came out here in the top talking about kind of an epidemiological study where they looked at pre-pandemic physical activity. So this paper, no biology, but purely just looking at this population of cohort of people, 61,000 men and women, 45-year older, and then looking at who had COVID, was hospitalized, who wasn't hospitalized, and then achieved metabolic equivalent hours, meaning exercising, running, or some kind of exercise that you elevated your level of exercise to a point where that's about 7.5 hours per week. So they actually found on their epidemiological data that indeed maybe this has improved the chances of a person getting, which might not be surprising to people, but lower the odds of the infection and hospitalization. So that was kind of this paper linked to that. There's this nice paper here that came out just a few days ago talking about the roles of vaccines and actually the protection role it has on cardiovascular compilations or thromboembolic activity. So it's a nice paper just showing that actually indeed the boosters, the vaccines actually lower your chance of COVID-related cardiovascular issues, which might not be surprising to people, but it's actually nice data to look at. But this leads to another paper here that came out again a few days ago talking about the virus-induced, basically how SARS-CoV-2 can basically really cause cardiomyopathy. Again, not probably a surprise here for people because we've been talking about it for a while. But there's some evidence here talking about the severe, next slide, severe infection to the lungs. But what's interesting here, they talk about how the immune system the macrophages can be involved, which could, again, go back to our inflammation paper and other things that are looking at with our group and immune-related factors, where they're basically saying, there's a cardiac where the severe infection to the lung will then actually be causing the cardiac macrophages to, in patients, to cause these spikes in inflammatory issues. You see, this is one of the figures here. They look like a bunch of inflammatory markers that we've actually shown in our paper too. But the interesting part of this paper is they use an anti-TNF therapy because they saw TNF alpha here that's up. And then when they use the anti-TNF therapy in their mouse model, they have the mouse model now they use after they look at the patient data. Oh, and the patient data, they actually compared 21 patients with SARS-CoV-2 with 33 patients who died of other causes, like hit by a car or something else, that didn't have it. But so the TNF alpha therapy actually shows all this reduction in the mouse model with all these inflammatory conditions that I think some of these inflammatory conditions are related to what we've shown in our papers too. So maybe this might be a potential therapeutic. So, this is an article from this NIH news release. I like this quote that they have in here from the author saying, this study shows that COVID infection and immune system can inflict remote damage, which we have already shown too in our group. But these findings are more general, kind of has, the kind of the impact overall impact over the whole body, which we also discuss a lot. This goes back to the spikes you might see in the wastewater. I'm not sure if Adam's here, but Adam might be the person to talk about this. But what they've seen is actually this paper showed that the JN.1 and the previous BA2.86 variants actually cause more shedding in the wastewater. So these spikes you might see in the wastewater could also be attributed to the JN.1 shedding more, which doesn't mean then there's more infections. It just means there's more shedding. So that's why you might see a spike in wastewater, which was interesting to see in this paper. They talked about it, which will cause other anomalies. When you're looking at wastewater, you might not actually, that spike you might see or thing go up to JN.1 might not be very reflective of the actual case numbers. It might be lower than that, as they indicate, which was interesting to see here. And this paper, I think, might be interested again to our inflammation group. Probably should reference this paper once our paper comes back from review. But they're talking about how ACE2 dependent independent SARS-CoV entries. This is their little model here where they looked at the inflammatory response and the replication. And there's a lot of overlap with what we're talking about here in our paper, but they have some other factors here that I think we didn't talk. But they basically focus on ORF6 and how that's involved with the early infections and then leads to the inflammatory response. That's kind of their take-home message. There's a lot more in this paper, but that's like a quick overview that I thought throw it up here that people might be interested in reading it in depth and see how things might be related. Again, going through this quick to give Cassian enough time. Okay, some recent long COVID news too. There's this recent paper that came out looking at long-term impact of psychiatric disorders. Again, no biology here, but here they basically looked at patients. I forget how many patients in the study they looked at, but and then they basically looked at the factor how many develop psychiatric disorders, from mood disorders to depression to panic disorders and so on, as you see here. And overall, which again might not be surprising for people here, but we see the increased rate of these psychological disorders that occur, whichever one you look at over time after you get COVID. And then this was an interesting article, which I'm not sure if people, I don't know too much about hypermobility. And the causes that I know hypermobility athletes have, and that's beneficial, but sometimes when you have hypermobility, it can cause joint issues and things like that. But for long COVID patients, this, again, epidemiologic kind of study, this big, they looked at all these patients and they saw COVID can cause basically, that there's 30% more likely that, for people who haven't recovered from COVID, that who have the normal joints will be more likely to recover quicker than the hypermobility people who have hypermobility cause the illness or not. So this is kind of an interesting little, again, no biology here. This is more of a survey they did, and they looked at people who had COVID, age, and so on.
I mean, I've heard of that. Actually, I didn't realize. Does COVID get into connective tissue, or it sits there? The SARS-CoV-2.
That I think people have shown it might. I'm not sure if it gets into connective tissue, but it does like downstream impact to biology. I don't know. Does anyone know the answer to Cassian's question? I'm not sure if anyone's shown it's connective tissue.
Seeing antigen not necessarily meaning it's replicating viruses
Yeah.
that are reported in synovial fluid.
I see.
But
Right.
I'm pretty sure I've seen something about that. Option maybe remember.
No, you're right, Steve. It does. Yeah. Not the replicating virus, but one of the exactly. Part of the viral particle. Oh, go ahead, Rob.
They've seen viral components in synovial fluid, but not evidence of replication.
Yeah, that's what my remembrance was as well. Yes, means
Rob,
maybe it was there once, but
Rob, you might know the answer to Cassie. Have you seen any kind of replicating virus in any connective tissue or no?
We haven't looked.
You haven't looked. Okay, that's the answer. We haven't looked.
That's good.
That doesn't mean someone else hasn't looked. It's just we haven't.
Yeah, I haven't seen. I don't think any papers. I think the thing I've seen is Steve. What you mentioned, you and Rob mentioned Steve. Yeah, so.
It's going to be an important, without belaboring, issue because so many of the rheumatoid arthritis-like symptoms and evidence for autoimmune profiles thereof are emerging in long COVID. It's a significant question.
Yeah, exactly. I agree.
Action.
Yeah, Bill.
Yeah. So a quick search reports brings up an NIH article that says SARS-CoV-2 RNA has been detected in sputum, nasopharyngeal secretion species in blood, but not in synovial fluid.
Interesting. Okay.
Yeah, but that's the particle. I mean, that's not the replicating virus, though. So that's what. Yeah. So we don't know if it is going to replicate. But yeah, thanks for the search. Yep. All right, I guess we'll go to Cassian with his non-favorite picture of himself. But
let me get
switch over to Cassian.
I want to share this as soon as possible here. There we go. Thank you. Okay.
Yeah. Let me unshare my screen. I think I'm unshared my screen there.
So thanks for inviting me, Afshin. Just as an introduction, we met at the meeting in Galveston through Space Science Summit, and I got interested in space science, strangely enough, not because I live in Houston. Oh, by the way, I'm probably the most politically disadvantaged individual here, I think. So just to be clear, I'm Canadian. Just because I live in Texas doesn't mean I support leadership.
We support people from all different
areas. I'm Canadian. Okay, so this is as far away as I can get geopolitically, I think.
Yeah.
But I can't remember. We just started talking about stuff, and I'm not even sure how COVID came up, except you mentioned these one of the meetings you were at, and so I don't. The interesting stuff that Ashton is talking about is actually near the end, so please don't manage your expectations. So here's the
Cassian, while you're giving your talk, do you want people to interrupt or wait till the end?
Oh, you can interrupt. Yeah, sure. I don't care. Yeah, it's fun that way. Make sure this is in the right place. Put this here. And as you're talking about, oh shoot, I need to swap, right? I think I need to swap.
You need to swap. Yeah.
There, and then as you brought this up, it's bringing a lot of micro PTSD because at that time we could not do any research. We had to do research in COVID. We actually got a supplement from the NIH. So amazingly, something good came out of that supplement in terms of research. So this is the lab, and these two people, actually I'm gonna, oh yeah, there we are, Yulin and Kay. did most of the work. They're in the back here, Yulin and Kirk. And then someone pointed them out. So this is the path: oncology, immunology, cell therapy, tumor engine discovery. Somehow ended up in SARS-CoV-2, and then now back in space science. But really, I'm an oncologist by training, immunologist by necessity, and then been doing cell therapy for about 25 years. And Steve and I have been on the can't remember. Was it Stand Up to Cancer? I think, yeah, where we first.
We've done that.
So here's the premise for this entire talk, which is what makes a good target for T cells. And since I'm an oncologist, this was all cancer focused. The first half of the talk, you're going to listen to about cancer stuff, but the principles that we applied were then applied to the SARS-CoV-2 epitopes. And so we'll skip that because that's adoptive cell therapy. So I know this is sort of cheesy, but you know, what makes a good T cell? And amazingly, actually, ChatGPT did a pretty good job. And this was actually the earlier version of ChatGPT. And so what T cells recognize through their T cell receptors, maybe most of you already know, is peptide presented by self-MHC on the surface. But what makes a good target? And they talk about things like foreignness, abundance, which I call peptide MHC density, stability, which you can interpret different ways. In the cancer setting, is tumorigenicity, right? Is that antigen loss or that peptide still presented by MHC? And then the important things is immunogenicity. Most importantly is immunogenicity. If something's not immunogenic, I would just say it's irrelevant. And there's a lot of quasi immunogenicity in the SARS-CoV-2 field. So hopefully I'm not offending anybody who might be in that camp, you know, the Alessandro Setti camp and all that. But I'm just gonna put it out there that first, so then, so then, no, foreignness is not necessary immunogenicity, just to be clear for all you neoantigen fans out there. Again, now that neoantigens, neoepitopes don't work, and they're very useful, and some have been even taken to clinic and cured patients, but that's not necessary, or is it sufficient? Abundance, of course, peptide immunogenicity I talked about already. And it's related to tumorigenicity in the cancer world, and I guess also, in the virus world, dominance. So I gave it an A-, it wasn't bad. Again, this is how a T cell recognizes tumor. Peptides drop on the surface. The self-MHC TCR sees that, and then it does its thing. Depending on the CD8, CD4 subset, whatever it is, it can release cytokines, so like granules, chemokines, what have you. CAR T cells, just to make this complete, recognize the surface antigen and therefore very limited in the scope of targets available to CAR. And the problem with the tumor field really is that you've got, oh great, happy guy, he's got a peptide, not that he's got a tumor, but he's got a peptide presented by his tumor that can be recognized by T cells. But 95% of these patients don't, unfortunately, have known targets for T cell receptors, even for most CAR. And the reason for this, CAR is the pure of the apple, surface available, not HLA restricted, which is a good thing. There's really only a handful of good CAR targets. And you've heard some news recently about, glioblastoma, fantastic news, CD19, BCMA, getting into the mainstream, but really very limited and largely liquid tumors. TCR targets, really only 4 or 5 here. But in fact, it's the core of the apple. It's the weight of the apple. It's where everything is, where the action should be going. Thousands of potential targets, which I guess, in my opinion, are largely being ignored because of the economic attractiveness of existing TCR targets. And so, and maybe the lack of will and courage to go after some of these other ones, which are not mutated, but still highly immunogenic. And so, whether it's virus-specific antigen or cancer antigens, the conventional approach, which has been in silico prediction, really is upon us. This was, just for those who remember history, was not the way the first epitope targets were identified. The first epitope targets identified was largely empiric fashion by Thierry Boone at the Ludwig Institute more than 25 plus years ago, when I started my career, which is basically he identified these empirically to show that the T cells recognize the tumor, and then he went after the target. Here, we seem to be doing everything backwards or forwards, however you want to look at it, because AI and deep learning and neural networks and so on have really taken over the field. And so there's a lot of prediction about immunogenicity, and these are the questions we need to answer, whether the SARS-CoV-2 or cancer antigens, is the epitope presented by the tumor cell, as I showed you before, by the self-MHC? Does it elicit a T cell response, and is T cell response of sufficient affinity to recognize tumor cells? Now I'm talking about natural TCRs, okay, not the ones that are affinity mature, which have their own problems. And then, of course, is the epitope presented with sufficient density to be recognized? And that is a problem with lots of HHV epitopes, for example, or the EBV epitopes, that they aren't present even though they seem to be immunogenic as a peptide. And then, of course, the ultimate test is transferring to TCR and showing you can transfer specificity and reactivity. And it's very difficult to prove just on face value whether an in silico predictive peptide is present. Now, these sequences and consensus motifs are getting better and better, and I'll show you the case of SARS-CoV-2 why that's a bit of a trap. You can't prove this unless you actually do the empiric studies. You can't just prove it in silico. And so what do? We do mass spec, basically pull the peptide, go where the money is, pull the peptide off the MHC, run your mass spec. And for cancer, this will be important because this is a database we use to analyze what I'll show you later. We have about 11 million spectra, so about 60 runs we've done with tumor lines. And we did convoluted that to 6,500 targets, which match all the criteria we discussed earlier. Are they tumorigenic? Is there some tumorigenic advantage to that particular protein or antigen from which the peptide is derived? Is it present in tumors we're interested in? Is it restricted expression, even though it's a self-antigen? So a lot of these are cancer testis, or cancer testis like antigen, especially in germinal or placental tissue and tumor cells. And then some of them are tissue specific, only expressed in breast tissue, normal breast tissue, malignant breast tissue, and so on, which make them actually good targets from a cancer standpoint. And then we winnowed it down to 250 really high value targets. And then once you do that, we go the next step, which is to generate T cells against these synthetic, make the peptides, synthesize them, and then generate T cells against the synthetic peptides and see if those T cells meet all the criteria we mentioned here in the middle here, which is do those naturally occurring TCRs and isolated from a broad TCR repertoire, able to recognize tumor or the target through the peptide MHC. This is very important. So what I said was not just recognize the peptide MHC, but also recognize tumor, and not just recognize tumor, but to recognize tumor via the peptide MHC, because this bystander effect that a lot of people seem to be ignoring. There's a huge bystander effect of T cells, again, which people are ignoring, and I can tell you that's a pretty robust bystander effect. And I think once you can meet all that, then you do your translation, basically, take the TCR transfer and see if you can recapitulate everything from nuts to bolts. And one thing I should mention here is that the reason why people don't do this, first, it's hard, it's difficult. Second, most people have gone to TIL, at least in the cancer world, to generate T cells against peptide epitope. And that's okay, because, you go where the money is, but problem is that very limited repertoire. We grow these from the peripheral blood. Healthy donors, patients, it doesn't matter. And the reason we can do this is because our 18 years of research in T cell therapy have been to generate T cells from peripheral blood of patients. And it turns out in the Science Immunology paper a few years back that the T cells that we generate, so just shortcut the whole story to the punchline here, is that T cells we generate from these patients from the peripheral blood and make them memory cells using some protocol which involves epigenetic stabilization. And we sort of dissected some of that with the IL-21 phospho-STAT3 scenario. But I think basically it is epigenetically stable, because once you make these memory cells, you put them into patients, they do retain all the memory features that you would expect. They become effector cells, revert to memory. So true memory cells. We generate those from the peripheral blood. We don't generate from TIL. And it turns out when we infuse patients with a uniform population, maybe 90% or so of these are antigen and tumor specific, each one of those clonal types you can then track and see, after adoptive transfer, how long they persist. But you can also go back and see if any of those clonal types exist in those patients before you treat them. And it turns out that you cannot find most of them. And the ones that do persist, those clonal types that you give to the patient persist, you definitely cannot find them in the patient's peripheral blood beforehand, which means that their frequency is less than 1 in a million, what have you. So very low frequency T cells we can isolate, fairly robust in vitro technology. And what that means is that if we take that workflow, we call that the ETC workflow, endogenous T cell workflow, apply it to this validation process, we are gangbusters in terms of, more than half, 70-80% of peptides we get, even though they're self-antigen, we can make T cells to those. And those T cells will recognize tumor. We haven't published this, but it turns out a lot of these are cell cycle proteins, kinetical complex proteins, things you don't expect to be immunogenic, but you can generate T cells and they kill tumor. Now we also got, some good ones that are a little more tumor specific, but I think, give you an idea, these are all the files and all the cancers we looked at, pretty broad spectrum, and actually some liquid tumors as well. And the interesting thing is that if the antigen is shared by RNA-seq or protein, the epitope is shared. And that's not always the case. That's not, one is not always equal to the other. You can antigen different epitopes presented by different tissue types. For example, you don't expect, liquid tumor completely different and ontology to express and process the antigen maybe to the same degree or level or immunogenicity as you would have solid tumor. But it turns out for several of these, the epitope is shared, and that's important because you're not always going to be able to do mass spec on tumors. And so, for example, some of these Co6A3, TOP2A, we got a few of these other ones, FANC1 and so on, they're shared, and that's why it's color matched here. Some of them more than 10 times across 5 cancer types or 3 times against 2 cancer types. So that's one unbiased view of it. The other is that the HLA prevalence is completely unbiased as well, because we're taking tumors from anyone and doing this mass spec. And it turns out, we cover more than 80% of the world's HLA class 1 alleles and a few others as well. Some, that are more common in Asia than they are in Western countries, but still pretty pan HLA inclusive. And so if we take a look, what we discovered is all the old known TCR antigens like MAGE4 and MIU, so even PRAME have very low transcript megabase expression, whereas the melanocyte-associated antigens are just a little bit better, but still between 1 and 3 transcript megabase, enough to call them immunogenic in this case. And here the CT antigen, this is normal tissue expression, fairly high. So this is what we have right now. This is what our peptidome now shows us. Lots of targets. This is just a partial list. And you can see all these antigens express different epitopes of different alleles across a number of different cancer types, and largely CT antigen restricted. So pretty good shared tumor associated antigens and then some breast tissue, prostate tissue, which are tumor specific. So what did we find? We found, for example, a lot of cancer testis antigens, but also found other types of antigens as well, tissue specific, as I mentioned before, proliferation associated antigens, and then some resistant genes like MDR genes and oral kinase A, which give rise to epitopes, which we can generate T cells and recognize tumor across a number of these different HLA alleles. So that's nice. This is great. It's pretty robust. This is an example of one of these. It's HORMAD1, actually. I can probably tell you what it is. A CT antigen been around for a long time, highly testis restricted, and lots of expression across a broad swath of solid tumors and some liquid tumors as well, and fairly high expression. Pretty good IHC staining associated with the EMP transformation, DNA repair. This is a great target. Found esophageal cancer, gastric cancer, A11 restricted, A2 restricted, lung cancer, and so on. So this is an example of an antigen's been around for a while. Everybody wants targets against CT antigens. Here's one that's been around for 10 years. Nobody has an epitope for it. And here we go. We got a T cell that kills very well, peptide dose titration against tumor as well. That's A2 positive, hormone positive. We make the TCRT even better. We don't even know where it drops off because now we're at 10 picomolar. Make the TCRT, kills tumor very well. I don't want to dwell on the cancer side of it, but I just want to show you that from nuts to bolts, you do the mass spec, you get a whole bunch of pretty good immunogenic targets. You pick the ones you want, and off you go. You're off to the races with this target.
Cassian?
Yeah, go ahead.
Is epitope binding affinities similar to insulin binding affinity to its receptor in that it's dependent on interstitial fluid pH? The lower the pH, the weaker the affinity, which leads to insulin resistance?
Jesus, I didn't even know about that. So it's true that if it's low pH, your peptide falls off. I can tell you that much. That's how we loop the peptide off the MHC.
Okay.
But the affinity of the T cell receptor, so that's the peptide to the MHC. But the affinity of the T cell receptor to the peptide MHC is a whole different story. Very difficult to predict, which is why we do things empirically. And so that affinity is probably more important than the peptide binding to MHC, just for your clarity. It is important, obviously, the peptide falls off and there's no target for the T cell to see. I totally agree with you. But predicting that affinity between the peptide and MHC, actually it turns out some of the weak binders are very highly immunogenic. because being resupplied, and then the TCR, as long as it sees it, it's an association rate. If it's slow, if it's for the TCR and the peptide MHC, then you get very high affinity overall. I know you're asking a different question, but I just wanted to make that distinction because a lot of consensus is based on peptide MHC binding, and your question may be more related to the tumor environment and the hostility for T cells to recognize that. Which
sure, thanks.
So I want to get to this slide really quickly just to get to the meat of it. And so here's some other targets. I'm going to skip through that, and we can do rare tumor antigens as well. Okay, 2019. We all know what that means. And this guy, he found the sequence. He was featured in Nature's Top 10 People and put it into the database. Fortunately, it wasn't discovered or followed a few days later. But then out of all that, people started making vaccines. And you know the story here. Along with that, though, was a lot of opportunity to publish papers. And most of them were people interested in looking for T-cell responses to the spike protein. Honest to God, I don't know why. Okay, I mean, I know that antibodies responses are important in virology and vaccinology, but why would you go after the spike protein from your T cell as the first thing you look at? Okay, I honestly do not know why. This one mutates. It's antigen loss, but that's what people went after. Why not look at everything else? Within the non-structural proteins, non-spike, and so. All these papers came out, Cell, MedArchive, Nature Neurology, and Cell Host Microbe, about 4,000 peptides altogether, and all predicted, okay, and across spanning all these class 1 and class 2 alleles. And it was very exciting because then people got on the bandwagon and said, we'll make peptide vaccines and adaptive biotechnologies. Oh yeah, we're going to make T-cell signatures for all this. Totally quasi-irrelevant because immunogenicity has not been demonstrated for these. And the way that immunogenicity was demonstrated for some of these was, hey, patients react to these, therefore it must be a response. And that's how they developed this paper here, which I think got published in some high-profile journal, saying, wow, we got responses against these 6 or 7 epitopes. Hey, and guess what? Those sequences also found in non-pathogenic coronavirus. And wow, how did that happen, right? Because these patients show up with these responses to some of these spike epitopes and spike proteins. Have they been exposed to these in the past and now they're reimmunized? What's going on? Okay, well, you're making all these predictions on the fact that this peptide first is processed and presented. So sort of missed first principles there altogether. What do I have here? Oh, here we go. So let's take a closer look. Let's take these peptides. Let's make them. Let's generate T cells against them. Here they are. All predicted, none eluded, A2 restricted. Okay, that's great. Wow, just by chance they happen to be A2 restricted. I wonder why that happens, because that's what we look for. If you look for it, that's where you find it. We generate the T cells, we sorted it, we expanded, very high uniformity, and I can show you the peptide dose titration, really good. None of them kill SARS-CoV-2 infected or expressed targets, even when they're overexpressed, none. So there's 7 of them. And the only one that kills is actually the 8th one, which turns out, I don't know if I have that data, which turns out to be the only one that was eluded off the peptide MHC. So, okay, let's pay attention to the other side of this. So to do this, we didn't have a BSL-3 rod. We couldn't grow out SARS-CoV-2. So we did the next best thing. We expressed the protein, many genes or long genes actually, of these areas, including spike member protein SP13. And then we had all these target cells. These are mostly tumor cells, but we used them as targets because we assumed that these tumors don't express coronavirus, obviously. Well, we assumed it's not a DNA virus. So we used that. And then we did the analysis, same as we did, except we used this as a starting point to load the peptide off the MHC and then ran it against the virus databases. And what did we get? We got spectra that we didn't deconvoluted, and here's an example of the first 5 that we published in PNAS. And you can see that the SARS-CoV-2, and they're partially conserved against the other pathogenic, non-pathogenic coronavirus. This is interesting. But more importantly, they are conserved across all the variants we had at that time. And then we've checked the latest one too. They're also conserved in all the variants. Even MGP, surprisingly, even though it's a structural protein. But all the non-structural proteins, these sequences conserved. And then so we did the business of generating the T cells and then seemed to recognize SARS-CoV-2 antigen protein expressing targets, and here's the first one, MGP65. We did the sorting, made the peptide, generated T cells, sorted, expanded. They kill peptide very well, but that's not interesting enough. But they actually do kill different, not just H375, H375, but ML624, a whole bunch of different targets expressing the membrane glycoprotein. They could do quite well. And here's E to F is actually the important piece of the puzzle, which again, very few people do, is to do cold target inhibition assay. So you can see here, hot target, meaning exactly what we see in the chart down below for chromium release assay. The T cell kills the tumor, or in this case, the SARS-CoV-2 expressing target is labeled with chromium. T cell kills, chromium is released, you measure the chromium, here's cytolysis, it's pretty good killing. If you add a cold target that also expresses that peptide, you're actually diluting the effect, you're distracting the T cell away from the hot target, and you get decreased killing. But you need your controls. You're just meaningless unless you have the controls. So if you add a cold target with an irrelevant peptide, does nothing, which is what's happening here. But and then if you add a lower dose of the relevant target on your cold target, relevant peptide on your cold target, you get a little more killing. So that's fine. So you see a dose titration. And so clearly this T cell recognizes this epitope of MGP65, so it's eluted off the MHC, and it kills this target in a peptide-specific fashion. We did the same thing for the other 4, and you can see here pretty good killing, dose titration. Most people just show you dose titration. Effective targets of 40 to 1, 20 to 1, but it's not bad. 10 to 1, 5 to 1, you still get killing. So we believe these peptide epitopes are real. We made the TCRT, we did the same thing. So what do we have? Remember I told you those first 8 so-called immunodominant epitopes published by many people, shown here, are represented in this table, and all 7 of them, which we tested and grew T cells on, don't kill SARS-CoV-2 positive targets. The only one that did among those 8 is the one we actually ended up eluting. And so of course, if it's predicted, yeah, you may get lucky, and some of them also happen to be eluted. And so that's the case here. That's why that one worked. We did a lot more work, and then we also looked at, hey, let's go for these other ones. And it turns out there's several that we identified by elution and are immunogenic, but also happen to be predicted. So in that pile, you get some bona fide peptide epitopes, but I can tell you most of them are not correct. And then here are some that were eluded but never predicted. And so these are the ones we think are de novo, and we tested killing, they all kill. Everything that's eluded kills. Things that are not eluded don't kill. So here's the question. Do these highly conserved SARS-CoV-2 epitopes make better vaccines? I don't know, because I kind of missed the boat here making peptide vaccine. But maybe to provide long-term protection, and maybe they can predict immunoprotection, from reinfection, relapse, or long COVID. Because I don't know, maybe somebody should look at that. We've been trying to get samples. So if anybody wants to, they're welcome to these peptides, run the assay, see if there's any prediction. But here's something that we did by accident. Remember I told you, well, we use these tumor targets because we thought, oh, well, they won't express coronavirus. Well, we were wrong. Turns out, if you just use the tumor, elute the peptide tumor, and then you run it against the that database, what do you get? I don't know. Turns out you get something interesting. I can share these peptides with you later. I didn't know the audience, but basically, this is the same peptide epitope that was eluted off. Sorry, I like that dog so much. It lost these 3 tumor targets: K562, which is the erythroid leukemia line; H1299, which is a lung cancer line. And we overexpress A3 and A11. So just to be clear, this may not be naturally physiologic, but they are there in this process and presented. Maybe it's the only way we could see the signal on the tandem aspect, which, as you know, requires some abundance of peptides to see it. But they came straight off of these tumors that were not infected or transfected or what have you with any SARS-CoV-2 fragment. And it's real. And the fact that it came off 2 different tumor lines and 3 different altogether. Oh, sorry, shoot. Did this wrong. That's what happens when I mess with the animation. Sorry. Okay, let's fix that. So then I asked Yulin to look even harder. I said, well, we got lucky. What about the non pathogenic coronavirus. Turns out there's two peptides with OC43 that match up and came off H460, which I think is also lung cancer. I can't remember. And then he went and looked even farther with the other coronaviruses and the SARS-2. I don't know enough about the genealogy. So not only K562 and what I showed you earlier, 1299, but also 1395 and H460. And I don't know, you guys can figure out what this means, but you know, some of it here, some of it here, it doesn't matter. It's coronavirus related. And so this is just a summary of what I talked about already. First 5 things, you know, I got on my soapbox and talked about that. These last 3 things is, you know, what does this mean? Why do tumor cells present coronavirus-associated peptides, or appear to? And do they elicit a tumor active? We don't know. Answer number 7. We should work on number 7. That's pretty important. And then if so, what's the clinical translational significance? I don't want to say that COVID vaccines are tumor vaccines, but maybe you can sort of try and appreciate what the immunity is. So that's it. That's all I have. Yeah.
Thanks, guys. Yeah. Then one thing I wanted to point out to people here, that one reason that the last part, I think we were talking about it, and we have a potential link that, you know, COVID could potentially cause cancers. And the report, Steve had introduced me to James Gregory in Colorado, Mercedes Riccone, actually had a paper that's about to be submitted that they're showing some really nice evidence that, and he presented here in December. But there's really nice evidence that SARS-CoV-2 can reactivate dormant tumors, not only SARS-CoV-2, but also flu virus to a lesser extent, but SARS-CoV-2 has a long life. So that's where when you told me about this stuff, so for background people here, about the connection of the epitopes with cancer, I was like, could be something there. I don't know what other people think, but you know, as you said, you left it there to be like the questions you had, right? The potential at the
end. So there's work to be done, but I think when you told me, I can't remember, so whoever knows this better, did you mention it was in certain tumor types in which the coronavirus was identified, or there was?
Well, no, the study that that James has done, that Steve knows, also familiar with, he did it on breast cancer specifically. So, and he hasn't looked at other. I mean, Steve, you correct me if I'm wrong, but I don't think he's looked at other tumor types yet.
Not to my knowledge. And Cassian, what he sees is he split patients initially before they did a lot of experimental work with breast cancer, who were doing well and didn't have any evidence that they knew of metastasis. But if they split them in fairly, you know, and they've extended this database between those they could well document it had COVID and those hadn't, they got a significant increase in the numbers of patients who had COVID where their lungs had metastases. And their hypothesis was that they're awakening dormant tumor cells that were there. And then they've done a lot of experimental work, which is very convincing, using an influenza model, and they're moving. So is that bulleted pretty well, Afshin? And it's a very deep study. They got about 14 supplemental figures and a lot of experimental data. And if it all holds, it's frightening. It's really kind of frightening. Yeah.
So wait, it's not you're not equating this with like a polyoma T antigen or like some virus-inducing cancer mechanism?
Well, they don't. I don't think enough knows there.
Okay.
It's an awakening of cells in patients that were already there. It's not implying a de novo process.
I see.
Creating cancer, although we talked a lot about that over the next 5 years, particularly for the bone marrow or lymphomas and other things, that MDS and other things that might show up.
I mean, one idea too, Steve and I went with him yesterday, talked about, is that, you know, it could be a de novo, categorized as de novo, but everyone has dormant tumors sitting in there. So that de novo could have been a dormant tumor, right? That got activated.
Freaking me out, happened. Yeah. Right.
But it could be related with your epitope story you have here. That's the part where the questions you left. I mean, I think, I don't know how, I guess you would need samples for that to prove it, right? From the long COVID patients, those cancer patients, is that what you need?
Yeah, so I guess the question was, okay, so where's the beef? Where's the RNA virus? Where's that fragment? should we be able to detect it in the tumors? And is that what that study showed, that they actually find sequences in the tumor being expressed, or was it just
No, they didn't look at that. I don't think, Steve, if I'm right, I don't think they looked at to see if the sequence is in the tumors. Do you know, Steve? Or no?
I don't recall any such data. If they have, it's such a voluminous amount of data we heard.
Okay.
I don't remember.
But they might have samples that then we can go back and look at that, because if they haven't looked at that, they might have access to the tumor samples from those patients that Steve mentioned.
So I don't know what to look for, actually. So we looked, oh, hey, we have RNA-seq for some of these tumors. Is the sequence there? Well, it's not the right, I don't know enough about molecular biology, to be honest. If it's not there, if it's not sensitive enough, you're not going to pick it up. Or if it's episomal, will you pick it up? So is there, what are we talking about here? Is there some heritable episomal, so that's why when it was there, we thought, oh, this is an artifact, this is something else, but we can't find that sequence in any other, database.
Very parenthetical question, I'm sure. So it's tumor cell specific, and you didn't see any of this activity in normal cells?
Well, yeah. So first of all, I mean, we look where the light is shining. We only had, whatever, those 40 tumors, and we saw it in some of them, right? Some of the spectra that was eluted, deconvoluted, and had the peptide come out. So we saw it in some of them. But we didn't look at normal tissue. We didn't do mass spec on normal tissue, although I'm sure there's databases we'd go back and see, if that exists or not. Now, keep in mind that, some of this is overexpressed class 1. So there may be tiny, tiny amounts, and that's enough to trigger a T cell response or trigger immunity. It's just that, I mean, for example, I just give you an example, MART1, which is like, highly immunogenic, melanoma, blah, 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 treat patients cured, blah, blah, blah. That falls off the peptide really easily, to address the point earlier, but it's highly immunogenic, and you don't pick it up on mass spec. It's very hard to pick up on mass spec. So it's possible when we overexpress this, we do see things that would normally present very, very low levels. Now, I don't know what that means. So I don't think mass spec is a good, general, you can make general conclusions about whether, it's going to be immunogenic or not, and whether it's going to be immunogenic in normal tissue or not. That would be intriguing. Yeah. Did that answer your question, Stephen?
Yeah. Eve, you're on, Eve, she studied a lot of open reading frames and analogies, and I can't remember. Eve, is it possible that you've seen all of this ORFs that are expressed in the human cells that he might be? I don't know. Maybe Eve can comment.
I'm not quite understanding the question, Steve.
So he's seeing that these tumor cells are expressing something that's recognized as the epitope from all the work he just talked about, and I'm wondering about homologous ORFs or something that's in human cells, and just whether you might.
Yeah, well, so there's a large number of ORFs that have evidence that have protein evidence in humans, and certainly it's possible. Nobody looks at it, so that's why we don't know.
So what's the best way to say, hey, is, because I know what the peptide epitope is, so we know what the corresponding nucleotide sequence is. What's the best way to say, hey, can you find it here?
As a transcript or as a protein?
Anything.
Well, if you know the sequence, you can search the RNA-seq evidence.
Yeah, okay, we can do that. Is that sensitive enough? Is that,
Well,
yeah.
How long is it, what you've got?
Oh, the peptides are only 10 amino acids, but they come from a gene which may be much longer. Yeah.
You could also, like, we have a bioarchives that we just released that has that a few of the people here are on, like Cassian, that has all the predicted ORFs from cancer patients and COVID patients.
Oh.
and non. Yeah, I'll put it in the chat. Okay.
Okay, thank you.
It's a pretty nice study.
Cassian, I knew she had this extensive.
Got it. Okay.
I mean, maybe Eve, I think maybe what Cassian's asking, like, Jeff have the pipelines or Ermi to do that stuff to look at kind of scan quickly?
Yeah, Jeff definitely does. has it also, but I doubt
He's
that
busy doing other things.
he's in Florida right now. He's really, yeah, but Jeff certainly does.
So
I mean,
there
it's
could be
a pretty simple
yeah.
pipeline. We could teach it really easily. It's not like a big thing, it's like really simple. It's just adding a bunch of sequences to what's normally already done. So yeah, I'm trying to get to the chat here.
Okay, well, I can, I'll ask my data scientist guy, Yulin, to take a look at that and work with you guys on it. I mean, so I don't know what to make of this, honestly. I mean, obviously, if we can't generate the T cells and we can't prove this peptide MHC is actually on the surface, this may be moot, or unless there's some other clinical evidence. I mean, we believe is on this. Oh, actually, you know what we need to run, actually, to be honest, I don't know if any mass spec people here is this is radio labeled or a heavy peptide, just to prove that peptide is there. We would probably do that first. And if it's real, then we can just publish that and people can do what they want with that data. I think that's sufficient data to show that that peptide is present. The question then is, if you generate T cells, can you generate T cells, recognize it, and so on, which would be of some, narrow significance from a translational standpoint. But the other question then is, what does it mean, physiologically, biologically? Does it represent anything?
I guess we have two things here, right? One is one of two things. We have a lot of things, but one thing is if we're able to give you this, let's say one thing is the samples from the patients that Steve described for our collaborator James in University of Colorado. If he has the tumor from the patients who got the activated dormant, and sent that to you, I mean that's something you could easily see. If
no, that'd be great. We could run the mass spec on those. But we need, I don't know. I mean, have lines been made, or were they just tumor samples that were frozen, or?
I have to ask him. He's done the whole mouse, as Steve described. He did all these extensive mouse experiments with standard tumor lines. I forget the MCF lines. I forget which lines he uses.
Breast cancer line. Yeah. Okay.
Yeah. But from the patients, I think he actually took samples to. Maybe I'll circle back with him. I'll introduce you guys.
Okay. Thank you.
Yeah. And then and then I guess the other part is like the screening that Eve said, and Eve just put the paper in the.
Yeah, I see that. I just pulled it up on my Thank you. Okay. I'll take a look.
Because that's the easy part for that screen.
Yeah.
Yeah.
So unless somebody, or maybe Stephen, I don't know, is there such a thing as episomal heritability, or what? Why would they even be there? I just don't understand how. Let's suppose this is true. Why are they present in these lines? Any
that someone else might have to answer that question. Steve or Eve or anyone else.
There's an episomal specialist on here. I'm trying to think of all the heritable sequences. They can carry almost anything, as you know, Cassian. So
okay. Okay.
So you're evolutionary. Maybe Nydia, you're an evolutionary biologist type person. I don't know if you have any clue. I'm putting Nydia on the spot. But
yeah. No worries. Cassian, do you mind repeating the question?
So why are they let's suppose all this is true. Let's say there's a peptide epitope. Let's say it comes from a fragment of coronavirus. Is it just integrated in the genome randomly, some of this, or how does it become heritable? Why is it even in my cell line?
Yeah.
It's an RNA virus, right? Obviously. So I mean, don't tell people that.
And I don't think I have much insight.
Yeah, sorry. If Nydia doesn't have the answer, I don't know who will.
Maybe tumors from patients who have been exposed to various infections. And the tumor maintains that. That's a possibility.
Okay.
I'm not surprised at, but so many of the cancers, as Cassian may well know, and it's very vivid in our inflammation studies in the paper. When you see the virus come into the nose and you look at the RNA sequencing, like many of the immune and interferon-driven genes, the MHCs are way up. They're way up.
Yeah.
If you look in the tissues of the patients who died, which is what our paper is about, it's completely the opposite direction. They're just lost. And as you know, that whole pathway from the TAP proteins and assemblies, everything can be hypermethylated concordantly and silenced in many cancers. Yet your tumor cells apparently have MHC that are presenting. Is that correct?
Yes.
That's interesting.
Correct. Yeah, that's absolutely correct. Okay. I think I understand. We have 3 minutes. I just want to get back to the other question, which is more real to me, is that we have all these T cell epitopes for non-structural proteins. Does anybody want to, are there any samples who I can access for immunity to see what those responses are? You're welcome to all 18, 20 of them. But we could do it. I don't care. I just need something to track that's clinically annotated.
I'm trying to think who here would. I know, Doug, you just got on, so you might not know, but Doug, you might have some sample or cell lines in the lab that you could maybe share with Cassian. I don't know,
well,
Doug.
It's you mean about? So are there PBMC samples from patients who?
Well, that would be Rob Schwartz. He was
okay,
on. He dropped off. So Rob Schwartz, I would have to. He was on here. So
that's fine.
You guys.
Okay, and then I'm sorry, Doug, was the question for the cancer line, the second part of the?
No, I just put Doug on the spot.
Okay.
Yeah, and I'm sorry, I had an administrative meeting I had to go to, so I just got off it. Sorry, I missed your talk, Dr. Yee.
That's okay, I can forward you the slides, but
that'd be wonderful. Yeah.
I didn't know which question we were asking Doug. So was the question?
No, I was saying for the samples themselves. Like,
the samples. Yeah.
So there's one thing we're getting. So just a background for you, Cassian. Well, I don't know if Doug got the approval paperwork yet from this group called EPIC from the Uniformed Services University. They have patient long COVID samples and acute COVID samples from patients who from the military, that been patients who had COVID or not, and then tracked it over time. And it'd be a PBMC or whole blood, whatever you request. So I know Doug's requested a bunch that he's gonna do mitochondria. Rob's getting some of that too, which I'm gonna talk to him. But that could be another avenue where you could get potential things, or look at that, because that's tracking from the same patient who had early on COVID, and then they developed long COVID in patients who did it.
Wow.
No cancer data on them, but it's a whole that. But you can still track that PBMCs or whole blood from that.
That'd be great.
Yeah.
Well, there's a new finding, Bill. Long COVID is similar to many other diseases we're finding now, like myalgic encephalomyelitis, chronic fatigue syndrome, as well as other persisting diseases following a wide variety of other infections and agents following major traumatic injury. So you may be there may be broadly available samples from all those communities, research communities.
Yeah, I mean that's one thing that Bill said. Yeah, the chronic fatigue syndrome and those patients, they've been showing good connection, but I mean, we think it's all really mitochondria because with the work we're doing with Doug, that we're all turned into mitochondriacs. But that's what
we are. We are. We're definitely mitochondriacs.
But that's another question for you, Cassian. Maybe we take offline is how does the mitochondria get impacted by this epitope changes you see in the cancers?
I don't know. You guys have to educate me. On mitochondrial implications. I actually don't know. Wait. Because
Doug has his hand up.
Doug, did you raise your hand?
Yes, we have been doing a fair amount of work on different mitochondrial and bioenergetic functions of different lymphoblastoid cell lines, and not cell lines, but lymphoblastoid groups. So like T regulatory cells are very oxidative, T effector cells are very glycolytic. So we have a joint paper with Martin Picard and colleagues where we've looked at all the different PBMC cell lines and done a complete biochemical mitochondrial workup. So if you'd like, if you email me, I can send you that paper.
Yeah, sure. Thank you. Yeah, that sort of shades into comes into our interest with memory cells and so on as well. Yeah, thank you.
Yeah, so I guess lots of stuff to follow up, but yeah, thanks, Cassian. I mean, we're at the top of the hour here, so I'll send emails to the people.
Okay.
If you don't have Doug's email, I'll send an email to you guys too. So
happy to share my slides. Thank you so much for the forum to present.
No, thanks, Cassian. No, we'll be in touch.
Great talk. Thank you.
Thanks.
Sorry, I missed
it. No worries.
So we'll get you guys connected, Doug. And
and you recorded this too. Yeah, so
I did.
Oh,
yeah.
okay.
All right. Very good.
All right, guys. Thanks, Cassian. Thanks,
Thank you, sir.
everyone.
Bye.
Bye.